The kidney is one of the most important organs involved in salt and water regulation, so in this final lecture, I'll tell you about how kidneys work in different kinds of animals. Generally, kidneys work by having a blind-ended tubule with a one-cell layer thick wall on the outside of the tubule, bathed on the outside by the coelomic fluid, the hemolymph, or the blood. Solutes in water are transported into the lumen or the open interior cavity of this tubule, either by transport mechanisms or by pressure, and then this filtered fluid flows through the tubule and either out into the environment, as you'll see in a crayfish, into the gut, as you'll see in insect Malpighian tubules, or into separate urinary tract. In vertebrates, the basic unit is the nephron. It's a hollow tube with a capsule at the end that ultrafilters the plasma. Salts, glucose, amino acids, everything except proteins, are forced into the lumen of the tubule. At this point, the ultrafiltrate is referred to as the primary urine. So the primary urine is what is filtered at the Bowman's capsule. This is then followed by selective reabsorption of the needed solutes, things you don't want to get rid of, like salts you want to keep, glucose, amino acids, etc. After all this modification of the ultrafiltrate takes place, what's left is called the definitive urine. So the definitive urine is what leaves the kidney and passes further down the urinary tract. Vertebrate animals have a very complex structure at the head of the nephron called the renal corpuscle. The renal corpuscle consists of two parts, the glomerulus and the Bowman's capsule. The Bowman's capsule is an invaginated structure of the tubule itself that surrounds the glomerulus. The glomerulus is an anastomosing arterial network of capillaries that's under high pressure. The capillaries of the glomerulus are in close contact with the tubule wall. The walls of these capillaries are fenestrated, meaning they have holes in them. There is then what's called a basement membrane, which is shared by both the capillary and the Bowman's capsule. The basement membrane acts as a sieve, as it has small holes in it. Podocytes are specialized cells on the tubular side of the basement membrane. They have projections that cross over to form a network which forms a type of filter. Plasma from the blood is forced through the filter by hydrostatic pressure, filling the tubule with fluid and maintaining bulk flow of fluid through the tubule at all times. This is a crucial point. There is constant bulk flow of new fluid being forced into the Bowman's capsule all the time. This pushes the urine down through the nephron. Let's look at a generalized vertebrate nephron by looking at an amphibian nephron. Urine flows through the nephron in the following sequence. From the Bowman's capsule, it passes through the proximal convoluted tubule, or PCT, then through an intermediate segment, then what we call the distal convoluted tubule, or DCT, the collecting duct, and then the ureter. The ureter carries the urine to its final destination. In fish, amphibians, reptiles, and birds, this would be the cloaca. The cloaca is a common area at the end of the digestive tract where the digestive tract, the reproductive tract, and the urinary tract all feed in together. Cloaca literally means sewer and comes from the Latin word used by the Romans. In some animals, most notably mammals, the ureters flow into a bladder where urine can be stored for later elimination. The primary urine flows into the PCT, or the proximal convoluted tubule. The cells lining the PCT reabsorb most of the ultrafiltrate that came in the Bowman's capsule, and most of the key solutes like glucose, amino acids, and a lot of salts. There is active uptake of sodium and active or passive chloride reabsorption. The PCT is also permeable to water, so that 40% of the water recovery happens here. This water and other solutes are taken up by associated capillaries that run along the outside of the nephron. The urine then flows down to the distal convoluted tubule, or DCT. There is active sodium chloride uptake back into the blood, but the water permeability of the DCT is adjustable, as I'll show you in more detail later. This is how the nephron is able to make a dilute urine. If you don't reabsorb the water, then the osmotic pressure of the remaining urine gets lower and lower. The urine becomes more dilute if the water is left inside the nephron while salts and other things are taken out. This permeability of the DCT is under hormonal control. This figure shows the difference in osmotic pressure and chloride concentration between the urine and the plasma in a mud puppy and a frog at different positions along the nephron and in the ureter. You can see that in the Bowman's capsule and along the proximal convoluted tubule, the osmotic pressure and chloride concentration is pretty much the same as you would find in the plasma, so it's isosmotic, and the UP would be 1. In the distal convoluted tubule, you can see that the osmotic pressure and the chloride concentration drops as sodium and chloride are retaken up, but water remains in the tubule. By the time the urine reaches the ureter, it's much more dilute than the plasma. This production of a dilute urine is called a diuresis, and under normal conditions in water, amphibians like this would produce a very dilute urine. The permeability to water of the distal convoluted tubule is under control of what's called antidiuretic hormone, sometimes referred in general as ADH. Antidiuretic hormones increase the water permeability of the distal convoluted tubule, leading to a concentrated urine being produced. In the case of amphibians, the most concentrated they would produce would have a UP ratio of 1, about isoosmotic. They're not capable of making an even more concentrated urine, as I discussed in the previous lecture. The effect of ADH is to mobilize aquaporins to the membrane of the cells lining the distal convoluted tubule. And so when there are antidiuretic hormones around, more aquaporins are mobilized and water is allowed to pass back into the blood. However, when antidiuretic hormones are low, aquaporins are not mobilized to the membrane and the water stays inside the lumen of the tubule. In vertebrate animals, there are 2 types of ADHs. Non-mammals use what's called arginine vasotocin, or AVT. Most mammals, however, use what's called arginine vasopressin, or AVP. The way in which kidneys of some types of animals can produce a highly concentrated urine is a great example of how form and function come together. The mammalian kidney is highly organized. The cortex is the outer region. The medulla is an inner deep region. The renal papilla is a small elongated section that extends into the renal pelvis. The renal pelvis is an expansion of the ureters that collects urine exiting the kidney and carries the urine to the ureters. It acts kind of like a funnel. The ureter carries the urine away from the kidney and in mammals connects to the bladder. This is a complicated figure from your textbook that you need to study well. The anatomy of the mammalian nephron is the key to its concentrating ability. First, let's look at the nephron on the left side of this figure. You can see the renal corpuscle with the Bowman's capsule and glomerulus, the PCT, the DCT, and the collecting duct, which are the same as I described in the amphibian. What's noticeably different is the long expansion of the intermediate segment that's called the loop of Henle. There are descending and ascending limbs of the loop of Henle, and they play different functions that you need to understand. There is also a thick and thin segment of the loop of Henle. This spatial alignment of the nephron is key to its function. So you can see that the urine flows down the loop of Henle, then back up before entering into the collecting duct. The blood that leaves the glomerulus then follows the entire nephron, forming a structure called the vasa recta. This is important for the reuptake of nutrients and water and salt that come out of the nephron. Notice that the blood flow starts and exits at the cortex part of the kidney. There are 2 types of nephrons that are found in mammalian kidneys. Some are called cortical nephrons. These filter, but they do not contribute to the concentrating ability of the kidney. They're spatially restricted to the cortex of the kidney. Medullary nephrons have very long loops of Henle that extend deep down into the medulla, and they're crucial for the concentrating ability of the kidney. The collecting ducts at the bottom empty into the renal papilla. How do we know that the loop of Henle length is related to the urine concentrating ability? Well, we can measure what's called the relative medullary thickness, or RMT. This is simply the size of the medulla relative to the whole size of the kidney. Here's a figure from your textbook that shows the maximum urine concentration versus relative medullary thickness of a variety of mammals. You can see as RMT increases, the concentrating ability also increases. In general, small mammals will have greater relative medullary thickness than large mammals, And desert-adapted species will have especially large relative medullary thickness and very high urine concentrating ability. On the other hand, aquatic mammals living in fresh water, like beaver, tend to have very low relative medullary thickness and don't produce a very concentrated urine. These photographs compare the relative medullary thickness of three small rodents. The rat in the upper left has a relatively small RMT compared to the other two species and a pretty small renal papilla. The gerbil and the sand rat are highly adapted for living in deserts, and they have a very large RMT and very long renal papilla. They have very long loops of Henle. The diagram in the lower right shows how the loop extends way down into the renal papilla and then returns all the way back up to the top before emptying its urine into the collecting duct. Why would this be the case? It's all because the loops create what's called the medullary osmotic gradient.